game. They read and stay current with all the journals, and they're out giving lectures across the world. These are the brightest people you want to be with. It's all about providing hope, and that's to me what the best part about Pedal the Cause is. So we've been a partner with Pedal from the very beginning. I can still remember my first meeting with Bill Coleman when he came up with this idea and talked about it. Absolutely, I said, speaking for Children's Hospital, I said, we really want to be part of this, and it's been great. I'm proud to say that personally I've been the team captain for the St. Louis Children's Hospital team for all 4 years. And I will also say that, speaking personally, the children that we sponsor each year have been an inspiration to me as a human being, and have made me a better person, and made me a better hospital president. First year was Victoria, the next year was Claire, the third year was Noah, and this year it's Ashley. Ashley and I just did a little photo shoot yesterday, and I was hoping that she'd be here tonight so that I could get her up here and embarrass her and do all sorts of things, but she couldn't make it tonight. Her mom's over here. I do have this beautiful picture of Ashley, so that's who we're writing for this year. Ashley was diagnosed with a large cell lymphoma about, not quite 4 years ago. It was 2009, late 2009. And here we are just 4 years later, and we are calling her a 3-time survivor in just 4 years. She's been treated initially, obviously, in 2009, successfully, then relapsed, treated again, relapsed, now treated again, and is in remission now for how long?
Over 2.5 years.
Over 2.5 years, and she's as vibrant. I don't know how we got her to sit still, actually, for the photo. She was all over the garden up on the 8th floor of the hospital yesterday. Just a bundle of joy, and a great joy to be with. So that's why we're all here. Just remember, it's to provide hope, to raise money, to fund research, to improve survival rates, and to provide hope. So to introduce talk a little bit more about Pedal and introduce our investigator tonight, I'm going to turn it over to the executive director who got this whole thing started after it was Bill's idea. This guy executed it and has put it on the map for us, and we're really proud to be partnered, as I said, with Pedal the Cause. And let me introduce now Jay DeVito, the executive director.
Thanks, Lee. And Lee is very modest. He didn't really talk about the size and the scope of his teams, but Lee has been a tremendous supporter from day one, and he has built pretty much one of the largest cycling teams every year. So, no pressure this year. We're counting on you again. But
we're 20 right now.
20. All right. Early bird ends July 1st. Don't forget. When Bill envisioned Pedal the Cause, it was, as Lee said, it was important to include St. Louis Children's Hospital. And I think that, it's been a great partnership, but I think that the partnership is best illustrated through our Ride for a Child program. And the Ride for a Child program pairs children that have been diagnosed with cancer and being treated at St. Louis Children's Hospital with a cycling team. And, while it's our mission to fund cancer treatments, fund cancer research with the goal of finding new treatments and cures, the Ride for a Child program really puts us very close to the human side of cancer. And we have witnessed some incredible things. In the last few years, we have seen some great inspirational stories of friendship and courage and as Lee says, hope and compassion. Clayco, our Ride for a Child sponsor, really led the way. When they adopted their child, Maddie, they immediately got to business doing what they do best. They built Maddie and her siblings a playground in their backyard to take Maddie's mind off her cancer treatments. We have Terry Greek here, who's a cancer survivor and one of the captains of one of our biggest teams, Powered by Hope. She would go with her child, Caitlin, for hospital visits and for chemo treatments. We have, Carissa Swigert, captain of the Sam Adams team, couldn't be here tonight, but she actually visited with her child, Nate, and her mom, Ro, in Philadelphia, when Nate was in Philadelphia getting treatments there. And then there's Dave Hill and the Hill Climbers. And Dave Hill and the Hill Climbers, Dave, raise your hand. This is a special story. You know, him and his team visited Seamus, their child, for months on end in the hospital, making weekly, almost daily food deliveries and offering moral support, because their family was from Springfield, and they were spending at least 4 or 5 months in Children's Hospital straight. So it was a great friendship, and watching it was terrific. You know, Petal is proud to play a small part in helping these kids and their families come through, you know, really the toughest time of their life. We were supposed to have a gentleman named Blake Adamson here. We have his dad Bryce here instead. Blake is at a practice, but Blake Adamson was one of our inaugural Ride for a Child kids. He was one of our first kids in 2010. And Blake is a healthy, you know, senior. He's a 3-sport athlete at MICDS, and he really represents what our ultimate goal is, you know, and that is to, you know, find an ultimate cure. But you know, it wouldn't be right if we didn't acknowledge the fact that you know, we experienced some losses too. You know, of our 22 Ride for a Child kids in 2012, we've lost 3 of them. And you know, Jamie, who's a 15-year-old, will be inspiring Team Revolution, an all-female cycling team who was very dedicated to it, and Jamie was described as a fighter. Seamus will forever be a hill climber, and you know, he'll be remembered for his kind spirit and his dry wit. And Claire will forever be remembered with Pedal the Cause. We've named our Claire Blaze Spirit Award in honor of Claire for her determination and passion in really getting after cancer research, building a team and fundraising, and really making a difference. And that trophy is so it's great to be here tonight to honor these kids and to also celebrate our successes. But clearly, we have a long way to go. And so it's my honor to invite Grant Challen up to talk about the award that the grant that he received from the Children's Discovery Institute, utilizing Pedal the Cause dollars to work on acute lymphoblastic leukemia, which is the most prevalent childhood cancer. So, Grant, thanks for being here, and it's all you.
Well, thanks everybody for coming out tonight, and thank you, of course, for Pedal the Cause and Children's Discovery Institute for supporting our work. And thank all of you in the audience for your time and your generous support of this great cause. And hopefully we can get going. Okay, so this is me. I'm a fairly new assistant professor at WashU here. I've been here for about a year. I'd just like to get started by asking for a show of hands in the audience for those individuals out there who've been affected by some way in cancer, either themselves, a family member, a friend, or anything like that. Should be most of you, hopefully. Okay, keep your hand up if that person who was affected had some type of blood cancer, leukemia, lymphoma, myeloma, something like that. Who do we have left? Okay, and then keep your hand raised if that person was a child. Okay, so we've got a good number here. For me, personally, my story started with a young man named Preston, whose family was a member of a church group I was associated with. And Preston was diagnosed with leukemia when he was 3 years old. And I got to see the impact that it had not only on him, but on his family and his parents, as he really battled the disease for a year. He ended up losing his fight. But it really made a lasting impression on me, the sort of impact not only on the child, but on the child's family. And it really gave me my first sort of intuition that I wanted to do something with my life to try and make a difference for these kids. And especially now that I have 2 kids of my own, a 2-year-old Matthew and a 2-month-old Michael, it really dawns on you the sense of responsibility for if something like this happens to one of your kids, and there's nothing you can really do about it, then it really is a devastating thing for a parent. So the project that we're funded by for Pedal for the Cause is investigating a disease called T-cell acute lymphoblastic leukemia, or TALL. So ALL is the most common form of cancer in children, and the 5-year survival of people diagnosed with ALL ranges from about 20% to 90%, and this varies greatly depending on the specific subtype of ALL that the child is diagnosed with. So we're interested in a particular subtype of ALL called T-cell ALL, which derives from the T cells in your blood, and it represents about 15% of cases in children and about 25% of cases in adults. And for some reason, this particular subtype of ALL has historically been linked with a much poorer prognosis and outcome compared to some of the other ALL subtypes, such as those derived from B cells. Just something I want you to keep your mind on is that over 50% of these cases of TLL have genetic mutations in this gene called Notch1. It's a very important gene for normal T-cell development. And most of you probably know that cancer is a disease that derives from genetic mutations. So changes to your DNA that then somehow change the function of that cell, that it doesn't do its normal jobs and it starts becoming a cancer. But in recent years, in science and medical research, we've really come to understand that cancer is not only a genetic disease, but it's also greatly influenced by epigenetics, and that's what we're particularly interested in. So most of you are probably out there saying, well, I know about genes and DNA, but what is epigenetics? So the genes in your genome contains all of the DNA, the sequence of nucleotides that tells your cell exactly how to function and really grow and develop everything you need in your body as you develop. So all of the information for making a human is encoded by your DNA. And essentially all of the cell types in your body have the exact same sequence of DNA. Now obviously a skin cell is very different from a nerve cell or a blood cell, but they all have the exact same set of instructions. So how are these different cell types constructed from the same source of information? And that information is encoded in what we call epigenetics. So epigenetics are chemical modifications or different marks that are made on the DNA by different epigenetic factors. And basically what this does is tell the cell which genes are going to be switched on and which genes are going to be switched off in a particular cell type. So epigenetics is really the instruction for the cell to tell it how to function properly. So the way I like to think about it is if you've ever bought a piece of furniture from IKEA or somewhere like that. It comes in this huge box, you open it and spilled out all over the floor a million little pieces, and you're left wondering, how am I going to do this? So all the pieces are the DNA. It's all the hardware you need to make the device. But without some type of instructions or roadmap, you're kind of lost. And that is what epigenetics tells the cell to do. It's really the instruction for how the cell functions. So the importance of epigenetics was highlighted in a recent cover article in Time magazine, saying that the DNA in your body is not only your destiny, it's also controlled by your epigenetics. And really, as researchers, we've come to understand that there really is a synergy between genetic mutations and epigenetics in developing cancer. So I'd just like for you to think of a particular genetic mutation as a fire in your genome. So when your cells decide to divide, they have to make an exact copy of the DNA strand from one cell to the other cell. Now, this process is normally very, very efficient, but occasionally there's 3 billion bases in your genome, and occasionally there will be 1 or 2 that are not copied quite correctly. Now, normally the cell has these sort of proofreading or safe mechanisms. Well, they'll recognize this error, which we call a mutation, and it will correct it before the cell finally makes another copy of itself. But sometimes the cells don't catch this, and these mutations slip through. And despite what you think about, this happens every day in everybody, in all of us, all the time. Now, for most of us, we will accumulate these small numbers of mutations throughout our life and it will never be a problem. Most of us will go on to have normal, healthy, long lives. But sometimes, if a genetic mutation happens in the right spot and it's surrounded by the right combination of epigenetic marks, then you really have a problem. And so I like to think of it as a genetic mutation is a spot fire in your genome, and the development of cancer depends on what epigenetics are around it. So if you have a fire in a desert, somewhere in your genome that's not very important and there's nothing much around it, it doesn't matter. You'll probably go on having a normal, healthy life. But suddenly, if you get a genetic mutation in an area of the genome which is important and is surrounded by the right combination of epigenetics, then you do have a big problem in cancer. And that's the analogy, if you get a fire in the forest, it's a lot different than getting a fire in the desert. So we study the epigenetics of TALL. And in particular, my lab has been working with this enzyme called DNMT3A. And it's one of the enzymes that's responsible for putting these epigenetic flags on the DNA. So there are genetic mutations in DNMT3A in about 20 to 25% of our TALL patients. And they're often found in the same patients that have the Notch1 mutations that I told you about earlier. So our hypothesis was that it's really the loss of these epigenetic marks, or the inappropriate placement of these epigenetic marks, that accelerates the formation of T-cell ALL in these patients. So our goal is to really identify ways that we can modify these epigenetic marks to help better treat these T-ALL patients, which have a much worse outcome to some of the other ALL subtypes. And the methods that we use in my lab are different genetic mouse strains. We have T-ALL cell lines that we grow in the lab, and we test many of our findings in primary T-ALL patient samples. So as I said, one of the major tools that we can use in the lab to study T-ALL is using different genetic mouse models. So we genetically engineered a mouse that the technical name is it's a conditional knockout of DNMT3A in hematopoietic stem cells. Basically what that means, in the stem cells that make your blood and bone marrow every day, we've made these mice so that they no longer have a functional copy of DNMT3A in their stem cells. So they're genetically deficient for the enzyme that establishes these epigenetic marks. What we then do is introduce the Notch1 mutation into either normal stem cells that have a functional DNMT3A or stem cells that no longer have this functional DNMT3A. And what we do after that, after we've introduced Notch1 mutation, is we take the normal stem cells or the 3A mutant stem cells and we place these stem cells into lethally irradiated mice in a bone marrow transplantation, and we monitor the outcome of TALL. And this is what happens. This is just one particular experiment. So the black line represents the survival curve of mice that were transplanted with normal stem cells that carried Notch1, and the red line represents the survival curve of mice that were transplanted with our mutant stem cells that had Notch1. So you can see that the control mice die at about 11 to 12 weeks from TALL, but our mutant mice succumb to TALL much faster, about half the time, in 5 to 6 weeks. This is what we call flow cytometry analysis of the blood, and this basically shows us the burden of the leukemic cells in the blood of the mice at the same time point. So this is at 6 weeks. We can see, I've just color-coded the red square is what you need to look at. That's the square where the leukemic cells are hiding, and you can see that there's a much higher burden of leukemia cells in the mice that had our mutant background. So this really tells us that the absence of normal epigenetic marks significantly accelerates the rate of T-cell acute lymphoblastic leukemia. So what we're working on now is really to understand why this happens, and we're doing many sort of molecular comparisons between our normal cells and the cells without the appropriate epigenetic marks. We want to identify the really specific differences in the epigenetic patterns and the genes which are affected by this from the Notch1 mutations. What we do after we've identified some specific patterns is we take our patient samples and we see if they have the same distribution of their epigenetic marks as what we see in the mice. And from our previous studies, we can say that if there's anything important going on in the mouse, it's almost always the same in human patients. So we're very confident about that. And of course, this is all ultimately working towards our goal of trying to come up with better therapies for these kids. So how can we restore or manipulate these epigenetic marks to come up with better treatments and better outcomes for these T-cell ALL patients? So that's just a very, very brief summary of the project. Most of you probably want to get home to watch Game 7 of the NBA Finals like me. So, as I said, I've been at WashU for about a year, and I have a small lab, but we're growing and recruiting all the time. I'd just like to acknowledge the people who've helped me in my lab so far, Andy and Kate, and of course the very generous support of our funding bodies, without which none of this would be possible. So the National Institutes of Health, Alex's Lemonade Stand, and particularly for this project, the Children's Discovery Institute, which was funded by Pedal for the Cause. So I'll close it there, and I'll be happy to take any questions either now, or I'll be hanging around a little bit until about 7 o'clock if anyone would rather approach me personally. So thank you very much for your attention.
Grant, that was great. We only have one more thing for tonight, and that's a short quiz. Okay, Carrie, can you pass out the example quiz? It's 50 questions, multiple choice. Grant, thank you. That was awesome. And Grant will be hanging around. Please stick around, have a drink, ask Grant any questions you may have. July 1 is our early bird special. We hope that you will let all your teams know. Thank you for being here, and have a great night. Take care.